Hi, everyone. My name is Clara Deck. I am with the NOAA Climate Program Office. This is our second webinar in our series highlighting C&GC Fellows, or the NOAA Climate and Global Change Postdoctoral Program. The C&GC program is sponsored by the NOAA Climate Program Office and managed by UCAR's Cooperative Programs for the Advancement of Earth System Science. Today, we're lucky to have Mukund Rao with us to present his research estimating the forest carbon cycle under climate change. Mukund is a current C&GC fellow and eco-climatologist who researches the interactions between climate change and natural and human ecosystems. He's currently working on projects related to the forest carbon cycle, the vulnerability of boreal forests to increasing heat waves, and the use of dendrochronology to understand past environmental change and timber transport. We will have time at the end for questions. Please go ahead and use the question box to enter your questions as we go along, or use the raise hand feature during the Q&A session so I can unmute you and you can ask your question directly. So without further ado, please welcome Mukund to begin his talk.
Thank you very much, Clara, and thank you to everyone at NOAA for the opportunity to present this seminar today. And I'm really excited to share some of the research that I've been working on over the past couple of years with everyone today. So there's two main topics that I want to talk about today, and both are related to forest carbon cycling, and particularly under climate change. In the first half of the talk, I want to talk about the critical temperature for photosynthesis. And in the second half of the talk, I want to make a case for why we really need to start considering tree growth as an essential part of the forest carbon cycle. So as we all know, we are emitting about 10 to 11 gigatons of carbon every year into the atmosphere, and forest ecosystems are taking up about a third of that. So that's the green line over here going into forest ecosystems, and the ocean is taking up another 25 to 30%. So these natural carbon sinks are offsetting about 50 to 60% of the CO2 emissions that we are emitting every year. So if the capacity of forests to do this in the future decreases, this means that there's more CO2 left in the atmosphere. And so we really need to understand the processes of carbon cycling in forests. So a few of the most important variables involved are photosynthesis, which is the process by which forests are taking up carbon dioxide, and in the process of doing this, they're transpiring water. That carbon then enters the forest. And trees then respire some of that carbon back into the atmosphere. Eventually, some of the carbon is accumulated as biomass that either is allocated to the leaves or is allocated to the trunk, both above and below ground. So it's a biomass that's really staying on the forest ecosystem for long time scales. That's when we're thinking about the carbon cycle, that's the carbon that we really need to be concerned about. There's other important processes in carbon cycling in forests, of course. So for example, there's herbivores that can eat a lot of leaves, and in some ecosystems, these processes like herbivory from leafcutter ants or goats can also be important sinks of carbon in the ecosystem. Now, when we're thinking about how carbon cycling in the forest is going to change, it's important to consider the fact that the environment modulates the processes of photosynthesis, growth, respiration. All of these processes are under independent environmental control. So the sensitivity of these carbon cycling processes can be different. And as I mentioned earlier, if we're thinking about forest carbon cycling over centennial or millennial scales, it's really the tree biomass that's the carbon that's staying on the landscape for really long time scale. So we also, instead of just understanding more about photosynthesis, we need to focus also on understanding the dynamics of how trees are growing and biomass is accumulated. Some of the challenges that we've faced in Earth system models for better understanding how the sink capacity of forests is changing or will change in the future comes from the fact that a lot of the Earth's current generation of Earth system models are formulated in a way that's very photosynthesis centric. So photosynthesis is the assimilation of carbon dioxide, and then once carbon is assimilated, it's allocated into these respiration and net primary productivity NPP pools. However, in most models, there's no independent environmental control of how the carbon is allocated into biomass. So you can have scenarios where if photosynthesis happens, the trees have to grow. Well, in reality, that might not necessarily be the case. And this is because if you're thinking just as a simple conceptual framework, photosynthesis can actually happen over a broader range of environmental conditions. Let's say warmer temperatures or higher vapor pressure deficit, then tree growth can happen. On the remote sensing side of things, we've typically used estimates of optical reflectance to monitor forest carbon cycling. And this is because forests are highly absorptive in the visible spectrum and highly reflective in the near infrared part of the spectrum. And this is because the plants are absorbing the visible light for photosynthesis. And while forests are absorbing visible light for photosynthesis, this does not necessarily mean that that visible light is actually being used for photosynthesis. And this is because there's other processes other than photosynthesis where this light energy can be diverted. So for example, in some ecosystems that are green throughout the year, if we were to look at metrics of greenness, such as the normalized difference vegetation index in a evergreen boreal ecosystem in Alaska, in the site in Delta Junction, or at a tropical evergreen site, we can see that the NDVI might remain flat throughout the year, but there's actually a strong seasonality to productivity. And in tropical ecosystems, this can be because of a wet season or a dry season, or a boreal evergreen ecosystems. This can be because of a summer winter differences. And so as I mentioned, once this light energy enters the leaf, traditionally we were interested in the reflectance that's coming back from the leaf. But once light energy is entering the leaf, it can actually go down a bunch of different pathways. There can be photochemistry that leads to photosynthesis. There can be heat dissipation or chlorophyll fluorescence. And the allocation of light energy to these three different pathways can be quite dynamic. However, in recent years, there's actually been pretty large advances in being able to track all of these three different pathways to really understand when are the plants photosynthesizing. Then another commonly used method to understand the forest carbon cycling is using the use of eddy covariance flux towers. And these eddy covariance flux towers are monitoring flux carbon and water and other fluxes at the ecosystem scale above the forest canopy. However, they don't really tell us much about the post-assimilation fate of carbon. So once the carbon is taken up from the atmosphere, is that going to the roots or the leaves or below ground? Those dynamics are not well resolved by eddy covariance flux towers. So most of my current research is focusing on understanding some component of these limitations that I've talked about. So the first part of the talk today, I want to talk about the critical temperature for photosynthesis. This is a paper that's currently under review. And in this study, we're going to focus on this region in northern Mongolia at the southern range of the northern hemisphere boreal forest. And so the olive shading here represents the range of the boreal forest in Eurasia and in North America as well. So most parts of the boreal region have warmed about 3 times the global average, and this is because of land atmosphere feedbacks and especially in further north due to polar Arctic amplification. So as the entire globe has warmed, and this figure shows global warming since 1950, and projections of future warming into the end of the scenario based on the range of CMAP6 scenarios. Northern Mongolia, which is the boreal region that we are focusing on, has warmed about 2 to 3 times the global average during the instrumental era, and it's expected to continue to warm at a rate about twice the global average towards the end of the century. And this is true both in this region, this part of the boreal forest, and boreal forests globally. So even if we were to follow a low emission scenario such as SSP1 2.6 for global emissions, we're expected to see much stronger warming in boreal regions than if we look at temperatures average globally. However, it's not mean temperatures, but it really is temperature extremes that are a driver of plant and human and animal survival. So if you're thinking about photosynthesis, this is the same conceptual figure I showed you earlier. Photosynthetic carbon assimilation is expected to follow these, or thought to follow these kind of optimum curves, where at low temperatures we have low photosynthesis, then we can reach some optimum temperature for photosynthesis, and then at warmer temperatures the rates of photosynthesis or carbon assimilation decrease again. However, in reality, if temperatures exceed a certain threshold known as the critical temperature of photosynthesis, which is typically between 40 and 50 degrees centigrade, this can cause leaf death. And it's important to consider these critical temperatures because leaf temperatures can actually exceed air temperatures by quite a bit. So leaves are absorbing shortwave radiation from the sun, and then leaves cool themselves by emitting some long-wave radiation, sensible heat flux, and latent heat flux. And this latent heat flux that comes from when the plants are taking up carbon dioxide and transpiring water, this latent heat flux is really important for cooling the leaves. However, there are scenarios where this latent heat flux pathway can be blocked, and this can happen during extended seasonal periods, during drought that can cause stomatal closure. And this also happens on a daily timescale in this phenomenon known as the midday depression of photosynthesis, where during the peak of the afternoon, because of high light availability, high vapor pressure deficits, the plants close their stomates during the middle of the afternoon. And both of these, both drought and the midday depression, can cause leaf temperatures to rise about 5 to 20 degrees centigrade warmer than the ambient 2-meter air temperature. So in this study, we use CMAP6 model scenarios and combine that with field-based measurements of the critical temperature of photosynthesis for Larix siberica, which is over here, and a range of other species in the region. And what we found is that for Larix siberica in particular, if we look at projections of future temperatures between 2050 and 2100, and the y-axis is the hottest leaf temperatures, under the two warmer scenarios, SSP3 7.0 and SSP5 8.5, we see that there's considerable overlap between the field measured critical temperature of photosynthesis for Larix siberica and what we're expecting in terms of leaf temperatures during heat wave events in the upcoming decades. And it's important to also note that Larix siberica is a really important species in the region. We're out here in northern Mongolia, we're at the southern range margin of the boreal forest, and Larix siberica covers about 80% of the biomass of northern Mongolia consists of Larix siberica, and about 1/3 of the Eurasian boreal forest biomass is Larix siberica contributes about 1/3 of the forest biomass for all of Eurasia. And it provides these important ecosystem services such as fuel wood and habitat for grazing of livestock. So in the study, what we found is that we're expecting temperatures to exceed the critical temperature of plant photosynthesis in the upcoming decades due to warming. And one of the larger takeaways is that we need to begin to include peak rate the parameterized decred in Earth system models. And this is because the current or most Earth system models today do not have an explicit representation of an upper temperature threshold for photosynthesis. So the way they're parameterized right now, as temperatures warm, the rates of photosynthesis decrease. But as if once you come back to cooler temperatures after a heat wave, photosynthesis can increase again. However, we know from field measurements and from many years of physiological studies, once the leaves are damaged, if temperatures exceed the critical temperature, on returning to cooler temperature, the rates of photosynthesis do not increase again. And this is because the plants, the thylakoid membranes and the photosynthesis system apparatus of the leaves have broken down. And so we already have lots of decred measurements for lots of species across the world. And I think that this is low-hanging fruit for us to include this information in our system models to better predict how the forest carbon cycle is going to evolve as the climate keeps warming. Clara, would you mind? Thank you. Perfect. Okay, so let's shift from the boreal forest in Eurasia to the eastern US, the northeastern US in particular. And in this study, that I want to talk about how our definition of the growing season, which is based on visual cues, might not necessarily be representative of the actual tree growing season. So here in this particular site, we're about 25 to 30 minutes north of New York City. And these are two photographs of an oak dominated forest, which is typical of large regions of the northeastern US and the eastern US more broadly. So it's about 80% oak. And we're looking on the left-hand side of a picture of the forest canopy early in the spring in April or so. And on the right-hand side, we're looking at the forest canopy in the end of October, early November. And so what we think of the growing season is based on these visual cues of when are the leaves coming out, when is the forest green, and when are the leaves senescing. And so we would think that in this particular region, the forest or the tree growing season would be April through October. However, growth is actually this invisible process that's happening at the micrometer scale, where you have these tiny cells that are dividing under the cambium of the trees. So if you want to understand when are the trees growing, we really need to be looking at this micrometer scale resolution. And so two ways we can do that is using these highly sensitive instruments known as point dendrometers, that are these spring-loaded sensors that can be affixed to the bark of the tree just about the living cambium. And so as the trees are growing, they can depress the sensor and we can get estimates of tree growth every hour or whatever the resolution of the instrument. Or we can also go out into the field and collect tree microcores every couple of weeks, as is what we did in the study, to look at the cellular scale of when are the trees actually growing. So what do the dendrometers tell us about the oak trees in this forest site? And so all the data I'm going to present for now are just going to be for the 2021 calendar year. The x-axis here is time, and the y-axis is the fraction of annual growth for 2021. And so these dendrometers were programmed to record tree growth information every hour. And we have 8 dendrometers on 2 oak species, red oaks and white oaks in the region. So what we see is that from January through April, there was not much happening. The dendrometers were not recording much growth. All the trees started growing around the 1st of April, and then grew really rapidly from April through the end of July or early August. And by the end of July, early August, by August 1st, the trees have grown about 84 to 97% of the total growth for the season. And then once we reach the end of August and early September, the trajectory of tree growth was flat and constant. So there was all tree growth was completed by the end of July, early August, with some amount of growth in August and then no growth through the rest of the year. So that was on the tree growth side of things. So now let's look on the canopy phenology side of things. And to look at the phenology of the canopy, we installed a phenocam that we programmed to take pictures of the forest canopy every 15 minutes. And I'm on the top of this image here, you can see the date and the time when this image was taken. And in the next slide, we're going to run through a GIF of what the forest looks like from January 1st of 2021 through the end of 2021. So we start off in early January. The trees you can see some snow. There's not much happening in terms of the forest canopy. And then as we get into early April, we're going to see that the leaves are just beginning to emerge in early April. And then by mid-May or the end of May, we can see that the leaves are completely green and the forest is green for this season. And the forest remains green through almost September to early October when the leaves begin to change again. So let's wait till we reach the end of August. Right now, the leaves are still green. September, the leaves continue to remain green. And then in early October is when we see the emergence of some fall colors and then by the end of October, early November, you can see peak fall colors, and then the canopy senesces by early November or so. And since we are actually right in the middle of fall right now, I thought I'd show you this image from this morning, October 31st, of what the forest looks like right now, where we can see these really nice peak autumn foliage. And that image, that GIF I showed you, can actually be used for doing scientific research, because all of those images, each of these images actually contain a lot of scientific information in them. So we can develop this or create this region of interest and extract the reflectance information in the red, green, blue, and the near infrared spectrum, and calculate vegetation indices such as the green chromatic coordinate or the normalized difference vegetation index. So the next slide, we're going to look at what the green chromatic coordinate look like for this particular forest, oak dominated forest site for the 2021 calendar year. From January through April, the forest, there were no leaves, so GCC remains pretty flat. Then in the first week of April or so is when the canopy just begins to emerge, and we can see this sharp ramp up as the canopy is greening, and we reach peak greenness in early May or so. And then we can see that the canopy remains green from May through the end of October, and the canopy senescence begins sometime at the end of October. And by early to mid-November, the canopy has senesced. So this is what we see from in situ remote sensing at the forest canopy site. And then we can also scale this up with what we see with what do the satellites see of the larger this forest site and the larger region. And the satellites can help us understand both how this fits in the larger context of the region, and also scale this to not just phenology, but also to gross primary productivity or carbon uptake by the forest. And so to do that, here I'm using the Tropomi sensor on the Sentinel satellites, and we use measurements of solar-induced chlorophyll fluorescence to estimate gross primary productivity of the forest. So the figure on the left-hand side here is going to be the same GIF that we saw the forest greening in the spring and senescing in the autumn. But now on the right-hand side, we're going to have a GIF of what gross primary productivity or carbon uptake of the forest look like during the same 2021 calendar year. So early on in the winter, there's not much happening in the forest. And in fact, actually, the forests are respiring a little bit. Then as we get into early April, we're going to see the forest GPP kicks in and the forest begins to absorb carbon dioxide from the atmosphere. You can see the green up is just about beginning to start. You can see this really strong GPP or carbon uptake through June, July, and into August. And then as we reach August and September, we can see that GPP values begin to decrease a little bit. Then once we reach October, we're back in the fall season and GPPs decreases quite rapidly. And then by early November, as the canopy senesces, there's no more carbon uptake. So we then average the GPP estimates for a region around our forest site. And this figure over here shows GPP from January 2021 through the end of 2021. Early on in the winter, there's not much GPP. And in mid-April is when GPP values begin to rise quite rapidly. We can see that there's this early peak in GPP in May. But the main peak of GPP where the most amount of carbon assimilation by the forest happens in the months of July, mid-June and July. And then once we reach September and October, GPP values have begun to decrease again as we enter into the fall season. So now let's put all of this together on, and sorry that the time frames have got cut off, but each vertical line here represents 1 month. So we start here in January and this should be December or the end of December or January. So let's put all of this information together. The top panel is for the dendrometers. The second panel comes from the phenocams. The third panel here comes from the tropomi sensor on the satellite. And the fourth panel here is from leaf level chlorophyll fluorescence data, where we calculated a parameter known as photosynthetic efficiency. So early on in the season, there's no leaves, so we cannot measure leaf level photosynthetic efficiency. And what we see is that there's no growth. GCC values and GPP values are flat. So from January through April. And then once we enter April, the trees begin to start growing. The canopy begins to start greening up, and eventually the GPP or carbon assimilation also kicks in. And for the photosynthetic efficiency parameter is an estimate of if you shine some amount of light on a leaf, how much of that light energy is going down the photosynthetic or photochemical pathway. And so the theoretical maximum for photosynthetic efficiency is around 0.8 to 0.84. And this is because if you're shining light on the leaf, it has to go into that photosynthetic or pathway or the chlorophyll fluorescence and the heat dissipation pathway. And so any values less than 0.8 means that energy is being dissipated as heat energy instead. So early on in the season, as the leaves are beginning to emerge, the efficiency of photosynthesis is suppressed by about 20 to 30%. And this is because the small emerging leaves are photoprotecting themselves from light damage or frost damage early on in the season as the leaves are emerging. So as we reach the end of May, around May 24th, that's when the photosynthetic efficiency reaches its peak for the season. And at this point, the trees have already grown about 25 to 40% of the annual growth for the season. So the first half of the tree growth for the season, we can see that this high growth is happening under a period when photosynthesis is not active or is not mobilized at the peak capacity for the season. Once we go into the months of June and July, we can see that both the trees are growing actively, the canopy is green, GPP is high, and the photosynthetic efficiency is high. And for me, what the most surprising thing that I found in this work is that once we reach the end of July, the trees have grown, almost completed the tree growth for the season. And so the trees are not growing between the months of August, September, and October, but the canopy remains green for 3 additional months in August, September, October, where there is GPP, even though it begins to decline towards the end of the season, and the leaf level photosynthetic efficiency remains high through the end of October. And so there's this asynchrony between what's happening on the tree growth side of things and the carbon assimilation side of things, where early on in the season, you can have tree growth with low carbon assimilation. And during the second half of the growing season, you actually have a 3-month period where you can have carbon assimilation without any tree growth. And we also then wanted to look at this at the cellular scale, because the tree dendrometers are only telling us about what's happening in the exterior of the tree. So we also went out and collected tree microcores every 2 weeks. So if you're not familiar with what oak tree rings look like, oaks have this really characteristic ring porous pattern where the early wood of the oaks have these large vessels that are used to conduct water from the roots to the canopy. And then in the second half of the growing season, the oaks produce the late wood of the oaks is formed with these much smaller vessels. So when we collected a microcore in mid-March, we only found the early wood of 2020 and the late wood of 2020. And this was the sampling was conducted in 2021 because that was the growing season we were interested in. And so we found no tree growth had commenced for 2021. As we moved to the next microcore in early April, we still saw the early wood of 2020, the late wood of 2020, and no additional tree growth for 2020. However, by the time we reached mid-April, we can see the early wood of 2020, the late wood of 2020, and a sliver or a band of early wood of the current 2021 growing season. And so that means the trees have already produced one band of the new xylem that's being used to conduct water. At the end of April, the early wood of 2021 is more fully formed, where you have that first band of early wood vessels is nearly complete. And by the time we reach the mid-May, we found that the entire early wood of the trees of the oak growth for that season had been complete. And so this can potentially explain why the trees are growing before photosynthesis is really kicked in, because to conduct photosynthesis, trees also are losing water at the same time. And you cannot conduct photosynthesis without having water available to the leaves. And the early wood vessels of the oaks, which are these large diameter pipes, are what are really important. They can help carry a lot of water to the leaves. And so the leaves are waiting till the early wood vessels are completely formed to then really kick in photosynthesis into high gear for the second half of the season. Then once we reach into June, the entire early wood is formed, and we can see the late wood of 2021 has begun to form. So the wood anatomy has already changed. By early July, we can see the full entire early wood for 2021 and almost the complete late wood for 2021. And by early August and September, we can see the entire early wood and late wood of the trees, and not additional growth does not happen once we reach into August and September. So to conclude, the main findings that I would like to conclude from this part of the study is that with these oak trees, we can see growth with low photosynthetic capacity early on in the spring and summer. And in the second half of the growing season, we see that carbon assimilation can continue without growth. And so the process of tree growth, tree photosynthesis and tree growth are not coupled. And so just knowing about photosynthesis from flex towers or from satellite remote sensing might not necessarily tell us what's happening on the tree growth side of things. That's the biomass that's remaining on the landscape. And what we are planning to do in this study is we take it further. We have a few additional dendrometer sites from colleagues in Illinois and some dendrometer data in Switzerland, and we also have a larger network of tree growth and from tree ring data across the northern hemisphere, where we find oak trees are broadly distributed in many regions of the northern hemisphere. So we are trying to test whether this is true across the distribution of oak sites that we have data for. So finally, in the last couple of minutes, I want to talk about some of the ongoing research that I have currently, and potentially invite some of you to collaborate if in the larger dendrometer flux tower network that I want to talk about. And so the overarching theme of my ongoing research is to look at the phenology of photosynthesis versus tree growth, and better understand the environmental drivers of photosynthesis and tree growth, to test the hypothesis of whether forest carbon cycling is source or photosynthetic assimilation, or sink, which is tree growth limited. And the idea being that or what we want to understand is, as we move forward in time and the climate is changing, which of these two processes are likely to be more important for the forest carbon cycle in the future. And to do this, we're developing this network of paired eddy covariance and flux tower sites, where we have eddy covariance towers that are monitoring carbon fluxes at the ecosystem scale. We have in situ remote sensing on the flux towers, and within the footprint of the flux towers, we are installing a network of point dendrometers that can help us understand the post-assimilation fate of forest carbon. So together we can, with the flux towers and the dendrometers and the remote sensing together, we can understand when is photosynthesis happening and when is tree growth happening. And then the idea is to scale this up using satellite remote sensing. And this figure over here that's overlaid on the Köppen climate classification map shows the current extent of our paired flux tower dendrometer map. And the base map over here can be deduced from combining the letter code for climate, precipitation, and temperature. So for example, the northern hemisphere poles correspond to the ET climate that corresponds to polar tundra, or maybe this region would be warm, temperate, deciduous forest. And so we've tried to develop a network that covers a range of ecosystem types. And using this paired flux tower dendrometer network at each of these sites, we're trying to test the hypothesis of when, or test and better understand the extent of the growing season for photosynthesis, and when are the trees actually growing at these sites. And we're actually adding a few additional sites, a blue oak site in the Sierra Nevada foothills next week, or actually this Thursday, and another site in Virginia next week, and another oak dominated site in Virginia, and hopefully a tropical deciduous site in Sonora in Mexico at the end of this year. So if any of you are running any flux tower sites, or would like to have tree growth information at your sites as well, please let me know, and I'd be really excited to collaborate. And with that, I'm also would like to thank the NOAA C&GC Climate and Global Change Fellowship for giving me an opportunity to travel to many of these sites to install the network of point dendrometers. And I'm happy to take any questions, and I'm also really grateful to a whole range of collaborators who have helped me in this work, and especially Troy Magny at UC Davis, who's been the host and postdoctoral advisor for this work. And I'd be happy to take some questions in the remaining time. Thank you very much. I am not sure I hear you, Clara.
Sorry, that's my fault. Unmuted now. Thank you so much. That was great. We do already have some questions coming in, so just a reminder that you are welcome to use the question box here. We can keep your slides up so you can reference them if needed. And then also, would you mind putting your email directly into the chat so that people have that as a link as well? So our first question is a question about the units of the photosynthetic efficiency. So is that 0.8, is that a percent? So that would be efficiency of 0.008.
So, sorry, that's scaled to 1. So 0.8 to 8.84 being the theoretical maximum means that if you shine light on a leaf, the maximum that goes down the photosynthesis or photochemical pathway is about 84% of that light. And anything lower than that, you can calculate if it's 0.6 and then theoretical maximum is 0.8 to 84, you can calculate that photosynthesis is suppressed by about 30%. But we also have other leaf level measurements such as non-photochemical quenching and light response curves that I did not mention here. So that backs up some of the photosynthetic efficiency data.
Great. Next question is, what is happening to the carbon that's assimilated without growth, root growth, or storage of sugars?
That's an excellent question. And so I think with the oak trees, there's this lagged effect where the photosynthetic assimilates that the trees obtain in the second half of the active season in August, September, October are what are cycled into the growth early on in the spring in the next calendar year when the trees are growing before photosynthesis is really kicked in. And so there's this lagged effect. And this has actually was observed even many decades ago in some of the radiocarbon data for oaks tree or radiocarbon and tree rings in oaks in Europe, where after the radiocarbon bomb spike, the oak tree ring data showed the spike in the early wood or with a lag of 1 year. And so the carbon that they got in the previous year was being used to grow carbon in the next, or for the tree growth in the next year.
Great. Next question is, do tree rings represent early growth wood or late growth wood?
Together, with tree rings, you can explicitly measure both the early wood and the late wood. And you can do this both using microcores that give you just a small sliver of the most recent growth, or you can take larger cores using an increment borer and look at tree growth in both early wood, late wood, or the total annual ring back in time during the entire course of the life of the tree.
Great. Next question is, nice presentation. This addresses overall forest growth, GPP. Don't you also have to look at NEE to get the net ecosystem productivity?
In this particular study, we could not, because we did not have a flux tower at the site, we did not have measurements of NEE, and the satellite remote sensing using SIF was calibrated to give us estimates of gross primary productivity. But what I want to do with the paired flux tower dendrometer network is to compare the phenology of GPP or photosynthesis, which is estimated from the NEE to NEE and to tree growth. But you're right that one of the variable that's maybe more linked to tree growth might be net ecosystem exchange or primary productivity. Thank you.
Another question, I know we're a little over time here, but if people want to stick around for these extra questions, I think we have a little bit of time to do that. So the next question, did you find any tea critical issues from a phenology standpoint during the unprecedented heat wave in June 2021 across the Northwest US and into British Columbia?
I have not, because these were I'm only conducted field measurements in Mongolia in 2019, but I would be really interested to see if people actually measured leaf temperatures and secret data during heat waves to see how close were the um, plants to exceeding their critical temperatures. And what I have seen when in forests during heat waves is you can often see evidence of sun scorch on trees, where some section of the tree, after a really severe few days of warm temperatures, one side of a tree can die off. Maybe it's the side that's in the northern hemisphere, and the southern facing tree that's more exposed to the highlight conditions, where the leaves also can reach warmer temperatures. So I think this is happening, but I think there's not been extensive studies that have really looked at leaf temperature, but there's been a few recently. The one that I can think of is this study in the Pacific Northwest that looked at consistently found using at a flux tower site in the Pacific Northwest. They found that consistently leaf temperatures were warmer than air temperatures at their site, and I think this was in PNAS.
Great. Another question, ecology focuses on energy balances. Do your findings of photosynthesis and sequestration cycles align with the ecological models of forests?
That's a hard question, because of course everything has to add up together, where the carbon that's assimilated from photosynthesis, it has to be used into either allocated to respiration or growth or other more transitory pools, but all of them have to add up in the long run. And I haven't focused on the closure of the forest carbon budget, but I think that would be interesting to look at it from that perspective. And I think the easier way to do that would be to use some kind of process-based model, because you cannot really measuring things like non-structural carbohydrates or shorter term pools in an actual forest is really hard. But I think with process-based models, that's something that can increasingly be done.
Great. Another question, thank you for the nice presentation. Have you compared GPP from the tower with the tree growth or LAI? Also, how did you measure leaf temperature?
The leaf temperature was measured in situ using a temperature probe. And to measure the critical temperature of photosynthesis, we actually used a fluorometer that was, we put a fluorometer on the leaf and warmed up the leaf and continuously measured fluorescence until we found this point at which the leaf fluorescence begins to increase exponentially, and that corresponds to the temperature at which the thylakoid membranes of the leaf were breaking down. So all of these ecophysiological measurements were conducted in the field. So it's the one caveat is that this was in a controlled environment in the field, so we don't have measurements of how often leaf temperatures were exceeding this in the field. The way that we did that was to use simulations of temperature and instrumental temperatures, but we don't have measurements of leaf temperature. And I forget the first question was about, the first part was about GPP. But
Yeah, 1 second. Sorry, I'm having some technical issues. Yeah, the first part of the question was Have you compared GPP from the tower with the growth or LAI?
And we are working on doing that for many of the sites.
Great. And one more question, I think today, how well is leaf drop assimilated into the landscape? Is this considered accumulation?
I'm not sure I understood the second part of how leaf drop is considered accumulation, whether it's recycled into the, I'm not sure I understood the second half of the question, but yes, there is a parameter in most search system models for the timing of leaf drop. However, I think our understanding of fall or autumn phenology is much, I think, more poorly constrained than springtime warming, because, for example, with the spring, we know that as the temperatures are warming, we have seen clear shifts with spring happening earlier in the season. But the fall phenology, there's an interplay of drought variability, or how was the summer conditions? Was it wet or dry? And warmer falls, all of these can play or come together. So like in some years you can still, for example, this year in the New York region, at least the fall is a bit earlier than previous years. And I think we still have things to understand about how fall phenology, the environmental controls and fall phenology.
Okay, great. And we have one last comment that this was a great presentation and the graphics were very helpful. So thank you again, Mukund. This was wonderful. Thank you to everyone who joined the webinar today. I put Mukund's email address into the chat, so please feel free to reach out if you have any other questions or interest in collaborating. It sounds like a very cool opportunity. So thank you again and hope to see you in our next webinar. Thank you.
Thank you. Thank you, everyone. Bye.